Today we will learn about arthropods of medical importance. So let's get started with a bit of classification. Now, Arthropoda is a phylum. It has medically important two classes, the class Insecta and class Arachnida. Now we will take one by one each category, each class. We will start with the Insecta class, and then later on in another lecture we will discuss about the class Arachnida. So here today I am going to discuss about the mosquitoes, which belongs to the class Insecta. So we'll discuss only about the mosquitoes of medical importance. There are plenty of mosquitoes in the environment, but we are not going to talk about any of those. We are only talking about the medically important ones, which are actually vectors of diseases. Generally, the mosquitoes feed on plant juices. Only the female mosquito requires a blood meal for their egg maturation process. So when you take certain species of mosquitoes, they prefer to bite humans, and some others they prefer to bite animals. Most of the medically important ones are actually zoophilic, which means they prefer to bite animals. They prefer animal blood over humans. But there are species which like human blood, prefer human blood. So, and their habit, the biting time could be either nocturnally or diurnally. That means they can bite you during night time or they can bite you during day. So, let's see the life cycle of mosquitoes. Now, mosquitoes have a complete life cycle, which means they have an adult stage which lay eggs, and these eggs hatch and release a larvae, and this larval stage become a pupal stage, and the pupal stage break off to release the larval stage. So you can see here in the picture, eggs, the larva, pupa, all are in the water phase, and the adult, actually the eggs are in the interface between the ground and the water. The larvae and the pupa are basically in water, and the adult is in the environment. So let's see what are the mosquitoes of medical importance. These mosquitoes species that we are going to talk about are the important ones for Sri Lanka. There are other species which are prevalent and important for disease transmission in other countries, but we are not going to talk about those. So there are two categories broadly divided into the mosquitoes of medical importance are divided broadly into two categories as anopheles and culicines. Now this culicine group contains the Culex, Mansonia and Aedes genus mosquitoes. Let's see the anopheline group first. The anopheline group contains the Anopheles culicifacies, which is the mosquito which transmits malaria in Sri Lanka, the main vector. If we take the culicines on the other hand, as I said, it contains Culex, Mansonia and Aedes genuses, so that we will be talking about each of these species which are prevalent in Sri Lanka. Now the C.quinquefasciatus and the Mansonia species are mainly important transmitting filariasis in Sri Lanka. The Culex is transmitted, C.quinquefasciatus transmits urban filariasis, and Mansonia annulifera and the Mansonia uniformis transmit rural filariasis. The Aedes aegypti and the Aedes albopictus are commonly known because they transmit dengue in Sri Lanka, and there are other Culex species like Culex gelidus and Culex tritriorhynchus which are important transmitting Japanese encephalitis in Sri Lanka, which are viral. So mosquito identification is our topic to go through now. So moving on to the mosquito. So let's look at the anatomy of the mosquito. Now mosquito being an insect, it has the head, thorax and the abdomen in a broad sense. So each of these structures have a significance in the identification, which we will look as we proceed in the. So when we want to determine which species the mosquito belongs to, the all four life cycle stages are important. That means even if we find an egg or larvae or pupae or adults, we can basically decide what the species of mosquito that is most of the time. So for example, if you take adult, the color of the adult or the mouth parts, the structures, the length and the various markings on the mouth parts or markings on the thorax, wings, legs, abdomen, or even their resting position could be helpful in determination of the species of the mosquitoes. So when we talk about the mouth parts, these are the mouth parts that we are talking about. We are talking about the antenna, which has hair on it, and then we have the proboscis, which is the structure in the middle, which is like a straw sucking blood, and then we talk about the palps, which are the structures which are on either sides of the proboscis. So based on the antenna, proboscis and the palps, mainly the antenna and the palps, we can sort of identify various species of mosquitoes easily. So let's see a comparison of anophelines and culisines. Now I said all four stages of the life cycle, the eggs, larvae, pupae and the adult can be useful, are useful in identification of the species, as well as I said the resting position on a surface is also important in terms of categorizing as whether they are anophelines or culisines, or even sometimes doing identification of the species. For example, if you take anophelines and the culisine group, you can see the Aedes. Now, anophelines have singly laid eggs, whereas the Aedes species also have singly laid eggs, but the Culex species, Culex genus, has eggs in a like a collection, so it's called a raft. So if you look at the larval stages of these two groups, again you can see in the anopheline group, the larvae is lying parallel to the water surface and the culicines are hanging on to the water surface at an angle. So that is the difference of the two groups. And if we look at the pupa also, they have various differences in the tuft of hair or the end of the structure, tail end of the structure, the shape of the structure. And then if you do the mouth parts, their males and females have their own differences in the antenna, and whereas palps have differences in the two groups, the anophelines and the culisines. And then I also said that the position, resting position, that means the anophelines usually rest at an angle. You know, if you take the body uh, line, they lie at an angle to the surface, whereas the culicines lie parallel to the surface. Their bodies line and the surface are sort of parallel to each other. So with these features, we can differentiate the species or the groups in the sense genus. So let's see the identification of adults. First thing is to see whether they are males or females. Now the males, if there are two mosquitoes, of course the males are usually smaller, but it is of course you have to have a lot of mosquitoes to compare. But then if you look at the antenna, the males have more hair on their antenna and the females have less hair in the antenna, with which you can understand whether they are males or females. This is the same thing which I have shown you in the diagrammatic representation in the middle, and on either sides you can see how it looks like under the microscope. Okay. So you can see the club shaped palps, and then you can see the short proboscis, short palps which are shorter than the proboscis. So with that you know which species and whether it is male and female. Of course male and female you can see by looking at the amount of hair on antenna as well. So moving on, let's say vector mosquitoes now one by one. So this is the Anopheles culicifacies which transmit malaria. Generally it's a zoophilic mosquito, which means it prefers animal blood over human blood. And nocturnal means they bite during night time. Endophagic means they bite you while you're inside. Exophagic means they bite you while you're outside. So which means Anopheles culicifacies, malaria transmitting mosquitoes in Sri Lanka, they can bite you while you're inside or while you're outside the house. And endophilic means after a blood meal, these malaria mosquitoes rest inside the house. So you can see them after a blood meal resting on the walls. So this is the Anopheles-culicifacies-morphology. These are brown color, light brown color mosquitoes. If you can see that at the upper border of the wing, it has alternating black and white markings. So with that you can recognize it as anophelines. And then the out of these black and white markings on the border of the upper border of the wing, the last one, the fifth one, the pale area is at the tip. Moving on, this is the breeding places that I have shown you in the picture. Now they breed in shallow, sunlit, clear, slow moving water. That means it could be, for example, I can give you examples like small tanks and ponds, or maybe shallow gem pits, or slow flowing water like the stream that I have shown in the picture, or even cart tracks can leave some markings on the ground. Those can collect after rain, those areas can collect some water. So when the water gets collected in those cart tracks, those are good breeding places for Anopheles culicifacies. Moving on, this is the Anopheles culicifacies eggs. Now generally they lay eggs in single format, like the eggs, each egg is separated from each other, and they have lateral floats. Lateral floats are on either sides. Now these help the eggs to float. It is just like the floaters that you fix onto babies when you put the babies into pools. Likewise, these eggs have lateral floats which help them to stay in the water. Now we are moving on to the C.quinquefasciatus, which is the filariasis mosquito. Now, filariasis mosquito also bites you during night time. They also can bite you while you are inside the house or while you are outside the house. And zoophilic means they prefer animal blood over human blood. And the difference with the malaria mosquito is that the Culex quinquefasciatus, this filaria transmitting mosquito in Sri Lanka, once they take a blood meal, they go and hide themselves behind wall hangers, screens, or certain paintings on the wall. Like they can hide themselves. They are not visibly seen like the malaria mosquito. So of course this affects the control programs, because when you spray uh, anti-insecticides, when you spray them on the wall, residual insecticides sprayed on the walls won't be much effective on the Culex quinquefasciatus. So moving on to the morphology, now Culex mosquitoes are dull brown in color, and they have no specific markings on the thorax, wings or legs. So how do we identify Culex species mosquitoes is with not having any positive signs on the body compared to the other species. So moving on, the breeding places of A.culicifacies are the highly polluted, organically polluted water. That's stagnant water, like the ones which you see, the drains which are blocked by certain structures or some way. If the drain is blocked, you can see that blackish water, which are with highly organic material and foul smelling kind of things. Those are the good breeding places for Culex quinquefasciatus. Moving on, the Culex quinquefasciatus eggs, they are generally in a sort of a collection. They are laid in a raft. The raft I have been given a picture here. That means their longitudinal angle is parallel to each other. So they are adhered or closely packed like kind of appearance is seen in Culex quinquefasciatus. We are now moving on to the Aedes species mosquitoes, which commonly transmit dengue in Sri Lanka. Now, there are two species which are commonly seen in Sri Lanka which are involved in the transmission of dengue. Those are the Aedes aegypti and Aedes albopictus. Now, these are day biters and they bite you while you are inside or while you are outside the house. And generally, these are very domesticated species of mosquitoes, which means they are kind of they try to stay close to the human habitat and stay inside the houses. And let's look at the morphology. In general, if you take both species together, these both species mosquitoes are medium in size and they are blackish mosquitoes, and they have a lot of silvery scales, giving like black and white dotted appearance. And these the markings are very much seen in the legs as well. Legs have bands as well. So these are the bands that I have drawn here or showing here using an arrow. And moving on, the breeding places of Aedes species, these are the temporary collection of water, like they are called container breeders basically, because they can breed in small collections of water like the flower vases or could be coconut shells or the tires which are left in the environment without filling it up with sand or whatever, or even leaf axils of trees or flower pots. Those can retain a small quantity of clean water. Those are good breeding places for Aedes mosquitoes. And generally these eggs are laid singly and they can stay for a long time, for a few months without getting killed by the heat or the temperature. Now these are some of the appearance of how the eggs look like. Generally the eggs are laid singly, and generally the Aedes mosquito lay eggs near the surface near the water, like if there is a it is generally fix their eggs onto the interface of the water and the air kind of place, and these are blackish ones and has actually a shiny black appearance and a smooth long oval in shape as well. Moving on to the species per se, now Aedes species, if we take Aedes aegypti adults, they have a lyre-shaped marking on the dorsal side of the thorax. Now, lyre is kind of the musical instrument that people say that Nero was playing while the Rome was on fire. That's the musical instrument that they have compared. Which is kind of seen on the dorsal side of the thorax of the Aedes aegypti. And moving on to the Aedes albopictus, they have a sort of a linear club-like marking on the dorsal side of the thorax, with which you can recognize them as Aedes albopictus. Okay, this is how it is seen. So moving on to another species, that is Mansonia species, which transmit Boogian filariasis in Sri Lanka. Now there are two species involved, Mansonia annulifera and Mansonia uniformis. These are all nocturnally biting mosquito species, and they bite you when you're outside the house. because that is because when we get to the breeding sites, you will understand why they are more on the outdoor biters. And in the adult morphology, they are light brown mosquitoes and they have heavily banded legs, and even their abdomen and legs both are banded. And wings have a specific appearance. They have like a pepper and salt appearance because of the light and dark scales which are nicely arranged over the wings, and these can be seen very nicely under the microscope. So this is the heavily banded legs, and on the other side you can see the wing. How does it look like under the microscope? So moving on to the Mansonia species breeding places, I said they are outdoor biters. That is because they breed in the undersurface of the water plants, like for example Pistia, Salvinia. Like they actually bite. They breed. They fix their eggs on the roots of these water plants. So this is how the eggs look like. They are in a cluster. The cluster is called a rosette, and then there are thousands of eggs in one rosette. And I said at the beginning that there are two species of Mansonia which are involved in transmitting rural paralysis in Sri Lanka. The Mansonia uniformis is one. Mansonia uniformis has a specific appearance on the thorax, which I mean, just to know uh, that it has a kind of a W marking on the thorax. And if you take annulifera on the other hand, it has 6 dots on the dorsal side of the thorax, and it's in pair format, two in a row. Okay. So now when you talk about mosquitoes, in certain instances when you work in entomological sections, departments, you will have to do certain entomological studies. Entomology is where you collect certain insects and study them for morphological identification. Let's see now these are the objectives of mosquito surveys. Now in Sri Lanka also we do a lot of mosquito surveys or entomological surveys on mosquito prevalent diseases. So these are the objectives of doing these. I mean, just for you to know what they are. Okay, I'm not going to details of them. Now, for certain for sample collection, we have different samples are collected. Like either at times adults are collected, or it could be the larvae which are collected, or even the eggs are collected for species identification and determination. Now, all the stages, even the pupal stages can be used, I said earlier also, for species identification. So the objective of sample collection is to determine the species mainly. Determine the species, plus here we want to see the infectivity, like the objectives. The first I listed down the objectives of doing entomological surveys. To fulfill those, we need to collect. Now these are few images of adult collection techniques where we have various trapping devices fixed. I mean, there are these generally people use baited traps. That means either a human bait, a human is inside the net, so the mosquitoes are attracted to him, or it could be an animal like a cattle kept inside the mosquito net, and sometimes they use double nets to protect the human or the animal inside, and then to trap the mosquito using a second net which is covering the first net. And there are other adult collection techniques like they use carbon dioxide to attract mosquitoes. There are vacuum pumps or light traps, so on and so forth. There are various adult collection traps and techniques which are used to collect adult stages of mosquitoes. And then larval collection also has different various methods. It could be using tipping technique, or it could be using a pipette where they just collect a small sample of water, like containing larva. For example, if it is a fall in a tree, then you can't actually do any dipping, or you can't put anything to anything else to get that water and the larva outside. So in that case, we can use a pipette. And now these are certain few indices that are commonly used when we refer up about mosquito collections and mosquito surveys, ecological surveys, like house index, container index, Brito index. So these are various terminology that you will come across just for your interest sake. So that's all. And please refer up the student seminar that we all did as well for further details of entomological surveys. Thank you.